Hello, everyone. Well, my name is Yassin Somoue Chafik. I am the current space major at the iGEM Foundation, and I am also one of the outreach coordinators here at the European Society of Synthetic Biology. The people that have organized these webinars and this festival. This is the first talk of the Space Festival, so I really hope that this introductory talk with the basic concepts of space synthetic biology can really help you. As a little bit of background, right now I am doing research on the pathobiome and how the pathogens can modify themselves in space, and how those conditions can come back to Earth and change the pathogenicity of the all of different microbes here on Earth. So I'm going to start my presentation. So in this presentation, as I've said, we are going to touch all of the different parts of space synthetic biology, and all of the we're going to focus mainly on the main issues that living beings that, don't have in Earth but do have in space, the different chassis that can be modified on Earth to fulfill synthetic biology in space, and some applications and some basic concepts in its policy. So let's start. Okay, so the first question that we should ask is why should we be interested in space synthetic biology? Well, as you know, right now we are having a lot of geopolitical problems, and we have seen that we need to start focusing on planetary resilience, since the resources here on Earth are starting to decrease. As a thing that we are going to see at the end of the presentation is that all of the research and all of the concepts that we have in space synthetic biology can come back to Earth. So everything that we discover in space can be translated into Earth, since here on Earth we're having the same scarcity problems and the same issues with resources. Also, we have seen that closed-loop life support is no longer optional, and that we need biological systems and engineering systems, both of them, that can be sustainable and that can replace themselves over time. As I've said before, we are with the strong climate crisis and planetary health concerns that we are having. We are in need of new technologies that can help us thrive in extreme conditions, such as space and here on Earth as well. Also, as advances in foundational advance. And something very similar is that biofoundries are enabling it, and the reports of Artemis 2 are going to be published in the next 3 weeks. And with those reports, especially in the Department of Life Sciences, are really going to help us to understand all of the constraints that we can find for example, in the Moon. So right now I'm going to talk about some key challenges on the field. So I have separated in 6 different branches. Okay, so all of the biological beings have different processes, and its physiology can be summed up in 6 different issues regarding space. So the first one is the microgravity. Okay, so microgravity has been is an enormous issue, because either in plants and in microbes, they can disrupt the cell growth, even more in bacteria, since the protein FINCZ is strongly to gravity. Also, all of the processes of biocontrol. So as we have seen, for example, the cellular membranes are subjected to gravity. So all of the mechanisms of creating energy in form of ATP in the membranes are disrupted in microgravity. So the integrity of the cell wall and the cell membrane is no longer viable, and it also alters plant tropisms in case of plants. Afterwards, if we focus on in cosmic and ionizing radiations, which are the main problems in space, we can see that, as most of my audience will know, is that the DNA is severely damaged by ionizing radiation, and a lot more in terms of 1 to the 14th way with cosmic radiation. Right now on Earth, we are protected by Earth's magnetic field and the atmosphere, which is not which is not a possibility in outer space. So the DNA is going to be very damaged, and the machinery to repair that DNA is going to be way more. It's not going to be viable, unfortunately. Also, it puts cells under cellular stress, and as I have said, it alters the healing machinery of the DNA. As well as automation and reliability, as you may know, is that cells are more even with time they need to be replaced, and machinery has to be replaced, which is the main issue. And you cannot take from Earth different electronical components which are going to be broken down with time. And the only way that in which you can transport important machinery is in having it in very small but efficient amounts, which is the case of for example, cell-free systems, and that with cell-free systems, for example, we are minimizing at the most the self-sustaining systems. When we are talking about biocontainment and planetary protection, we are going to see afterwards that the Outer Space Treaty of 1967, the COSPAR, and other small laws that are being applied to space are very specific to space, and we're going to see how outdated they are. Afterwards, we can see that we've got some issues with closed-loop biosystems. So, as you know, with resources, which is the main issue in space, that you don't have access to unlimited resources. And well, in Earth they're not unlimited, but you've got quite a field in which to play. So the first one is that you have got, as I've said, limited access. And the problem about the conditions on space is that you've got extreme pH and extreme salinity levels with high levels of heavy metals, which is, for example, the case of Mars, and that gives us the necessity of modifying organisms using biotech and genetic engineering to overcome these issues of extreme salinity and extreme cellular stress. And then we've got the extreme conditions in an abiotic stress, which is my field of investigation right now with the pathobiome thing, is that the temperature has got extreme fluctuations. For example, when you are focusing on Mercury, the side facing the sun is nearly 1,000 degrees, while the part behind the sun is at minus 100 degrees Celsius, which is a huge comparison between day and night. I think that Mercury is the biggest one. Or, for example, you can see when you're focusing on Venus. Venus has got one of the most is the hottest planet on the solar system. That gives us the immense possibilities on how it would work in all of the different exoplanets. Well, as I've said, you've got the vacuum, desiccation, the high concentrations of unwanted gases and heavy metals, which all of them are different resources which life on Earth is not adapted to, and simply because life on Earth did not evolve to work on that way. And now I'm going to talk about the different biological systems and chassis when we are working with synthetic biology. So, as I've said, well, all of these are the key challenges on the field. So we could sum them up in extreme conditions, microgravity and cellular stress, ionizing radiation, the need of economizing in logistics, all of the problems with biomanufacturing, and the resources. And now I'm going to talk about the different chassis that we have. Okay, so the excellence of synthetic biology on space are self-free systems. Basically, I assume vote here because they are non-living systems, so you don't have the need of taking into account the survival of these systems. They are super predictable. It has been seen that the coefficient of determination in experiments with self-free systems is super close to 1, and as we are going to see afterwards with mammalian, when we are working in space, is not close at all to 1. We are ranging between a 0.6 in R squared to a 0.8 You are not, while self-free systems are very close to 1. Sorry, and the support that you need is very limited. The bad thing about self-free systems is that the replicability is very, very, very low. Afterwards, we are followed by bacteria with the silver tier, which are easy. I mean, bacteria, as you already know, for example, E. coli you can have colonies at the next day at 27 degrees sorry, at 37 degrees, and afterwards it's very scalable because you can have lots of experiments in a very short period of time, just taking a very small aliquot. And by having different strains, you can modify you can modify bacteria to have different experiments and to have different outcomes. The bad thing about using bacteria is that the environment, as we have seen earlier, space between the conditions, the ionizing radiation, on the different between the conditions, the resources, and the radiation, you can have lots of mutations, and those mutations are the exact same thing that are going to modify those bacteria, and you will have non-viable bacteria, of course, because they won't be able to survive, or you will have bacteria that mutates a lot, and you won't understand the outcomes, which is the case, for example, of the pathobiome, I think. And afterwards, you've got yeast and fungi that are very robust because you are already starting in eukaryotes. There are model GMOs, as well, for example, as we're going to see with plants. And the good thing about yeast and fungi is that they help us a lot in biomanufacturing, which is like the main issue that we have on space, because when you're working in situ resource utilization, which is an application of space synthetic biology, which is mainly using the things that we have on space to give them a second use so we can survive on Earth, on space. And so they are very useful because yeast and fungi, as they are eukaryotes and they have stronger genomes, they can produce way more proteins and they can produce way more secondary compounds from its secondary metabolism, and that we can use, for example, in biomaterials and such. The bad thing is that as they are eukaryotes, I think that you're going to see in all of the eukaryotes right now, is that they are very slow. They are very difficult to modify, and they can mutate very easily. Afterwards, we are introducing ourselves to algae. So as the good thing is that they are photosynthetic. So as long as you have enough photosynthetic active radiation, the power light, you are going to be all right. They are very useful for in situ resource utilization and closed loop biosystems. The good thing also is that they are model GMOs, and for example, in the case of Chlamydomonas renarti, that we know all of the information, and you can modify them very easily. The bad thing is that they are very slow, as I have said with all eukaryotes, and the issue with algae is that they are especially sensitive. For example, when you want to modify duckweed, you have to be very, very, very careful because they are going to die super fast. But the good thing about algae is that they multiply their volume. In just 72 hours, we've got the double of biomass. Going to plants, which is one of my main focus of interest, is that they are very broad spectrum. You can use plants as biofactories. In 2023, I participated in a team that they wanted to use plants, especially tobacco plants, to produce insulin. So with a hand blender, you could blend those plants to gather that insulin for astronauts, and that can come back to Earth. And you can use that method to give insulin to people in middle and low income countries. And as I've said, the regulation with plants is super complex. Well, right now, as you know, the EU has changed the regulation regarding plants. So we hope that changes in the future. And also plants are very sensitive to mutations because they are more sensitive to light. And at the end, mammalian synthetic biology regarding space is the worst. You've got a very low predictability and very low correlation with your experiments. The good thing is that there are in-Earth models, so if you understand what happens with mammalian cells, you can understand what's going to happen with humans in space. And they are very good for testing. You can test mammalian cells to understand what will happen to our cells in microgravity, vacuum, in prolonged periods of time. But the bad thing is that they are super complex, they're non-scalable because it's very difficult to duplicate cells from humans or from mammalian tissues on space because the mitosis rate drops by a lot, and you've got a lot of biocontainment issues. So the ideal checklist for our chassis, it should be that we should have minimal genomes. So you should have genomes that are very slow or invisible, just like the case of bacteria and cell-free systems, which here is the problem with all eukaryotes. The good thing, you should also have evolutionary stability, which is something that we have that is quite an issue with all of the living beings. It's not the case with cell-free systems. You should have modular pathways. So in case of space, as you know, in the transcriptional unit that you have, you should be able to change all of the subgroups of the transcriptional units as Legos, so you can take advantage of all of your resources in a short period of time and in a very limited place. Imagine that you are working in the most basic lab, so you should be able to change very easily the transcriptional units to your favor, which is the case of cell-free systems. And afterwards, fault tolerance, and you should have low water and energy metabolism and needs. That's why plants and mammalian cells are in the lowest, because they are very complex and they are very needy. In those cases, they should have autonomous regulations, which is a little bit of an issue with all living beings. It should be extremely important, I'm going to stress this a lot, to have kill switch friendly systems. You cannot encourage scientists to modify things in space and have the risk of those mutated things coming back to Earth without the certainty that they are not going to create a damage here on Earth, and that it should be replicable, which is the case of all of them instead of cell-free systems. And so I'm going to show you some examples in nature. So as you know, evolution. I'm going to grab real quick my charger just in case. I am over this. Sorry. Okay, so as you can see, nature is very wise, and we have seen that synthetic biology. And as you know, synthetic biology, we use the things that nature has given us, like proteins and such, to be able to modify these organisms. So most of the genes that we have taken for synthetic biology in space are from the following. The main ones are from the following living beings. So you can see Cryptococcus neoformans, that they have a high capability of forming endospores. Thermus aquaticus, which we have taken from its genes to tolerate the high temperatures as an extremophile. Deinococcus radiodurans, which is amazing for radiation, and you've got Bacillus, the genus Bacillus for the spores. Here you've got them like with green malachite, and as you know, all of the tardigrades, which are also known as water bears, that we have used right now. They are being researched on how animal cells as testing facilities are going to tolerate stress in space. But why space synthetic biology? And here I've got some fields of study. So as I've said, we've got in situ resource utilization, which is using the things that you've got on space to create an environment, to create a human friendly environment. You've got afterwards life support, which if you have watched The Martian or Project Hail Mary, is using synthetic biology to create systems that can support us in agriculture, in medicine and such. Space biomining, which is very, very close to terraforming, is modifying organisms genetically so they can break down the exoplanet's lithosphere, and we can use those precious metals. And afterwards goes cybernetics, space manufacture, which is basically that, creating from fungi and such, with their secondary metabolites, things that we can use as biomaterials and such. Spatial medicine, which, as I said before, the use of creating medicine and all of that. And afterwards, we've got, very important to know, responsibility, ethics, and biosecurity. And here I've brought two examples of in situ resource utilization, because I think that it's from all of the niches and fields of study, because I mean, all of you can more or less understand all of this, but this is something very specific of space. So the first one is, well, these are two cases of study. The first one is something that I came up with to show you, and the second one is something really that is being done by the European Space Agency. So the first one is the production of oxygen using CO2. So at first glance, it looks just like a photosynthesis, but you have to modify that photosynthesis so it can be done on space. So the idea of this experiment that anyone can take care of is using photosynthesis to convert Martian CO2 in usable oxygen that can be breathed. So it would consist of modifying cyanobacteria to resist radiation and low pressure, optimizing its metabolism. Because as I said before, when you're using cyanobacteria and algae, you can duplicate it's like using all of the good things of plants, but without using the bad things of plants they take a lot to spawn. But cyanobacteria and algae in general, they need only a couple of hours. And it would be a great importance for closed-loop synthetic biology, because from starting you are using the resources of Earth, of space, in this case Martian CO2, and you are turning it into something that you need, which is oxygen. And that would create a loop, because afterwards humans and its infrastructure would breathe that oxygen, and then we would create more CO2, which would start a loop. And the second one is an example of in situ resource utilizations and biomining, that is extracting metals from regolith. Regolith is all the soil and the lithosphere of exoplanets. So they used Acidotiobacillus ferroxidans and Leptospirium ferroxidans, which are bacteria of extremophiles, sorry, and they use microbes to extract these precious metals by deposition on rocks. So it's part of the of ESA's BioRock project, and they optimize the metabolic routes to dissolve minerals faster and under low gravity. So they dissolve these minerals. So for example, there is a strain that is more focused, let's say, on cobalt, and this strain is going to dissolve all of the cobalt, and afterwards, by deposition, you can extract that cobalt very easily. And I'm going to talk very fast about biosecurity, because at the end of the month, you've got a talk with EBIS, one of the biggest foundations of biosecurity internationally. So I'm going to give like very quick notions. So biosecurity and planetary biology, we've got two main laws, which are the Outer Space Treaty of 1967 and COSPA. The Outer Space Treaty of 1967 is the only thing that is legally binding, and it's not just for synthetic biology, it's for anything related to space. And it's very outdated. I'm very critical with the Outer Space Treaty, because there are only two articles that are directly liaisoned with space synthetic biology. Some of them are very ballistic, some of them are more for engineers, most of them mainly, and for wars, but there are only two that could be directly impacted. That is that all sovereign nations that sign this treaty should not pollute the environment, and are responsible and liable for the pollution that they do. So the main issue with this thing is that I'm super critical with this one, is that since 1967, 60 years ago, nothing has changed. Nothing has changed. Since the ratification of this agreement, nobody has tried to create revisions and such, which is and in the field of deep tech, where everything is evolving super fast, we need a stronger regulation, because right now space synthetic biology is evolving fast, and its regulation is staying very far away. And here we've got two concepts that are forward and reverse contamination, which is the main thing, for example, in pathobiome. So that's why when we are talking about pollution and environment, the main issue is that forward contamination is that you cannot take anything that could pollute space and take it from Earth, for example, microbes or pathogens, take them, for example, to Mars, and pollute Mars with your pathogens. And the second one, reverse contamination, is taking anything that can have evolved on outer space and bringing it back to Earth. So you should leave them as two separate compartments. And this is in order to preserve scientific integrity, because if you are introducing different new strands of DNA in one of both of those compartments, you are disrupting the study of other disciplines regarding that place. So you should try to do research in separate compartments. And afterwards, we've got COSPAR, which is, I think that it's the, this is, I think that it's just an excuse to not have reviewed the Outer Space Treaty in the last years. This is very new. It's made by the panel of planetary protection. It's not legally binding. There are some indications to not to preserve fundamental research, as I said before, and to protect the Earth. It's very ambiguous, and it's very, there are more than rules. There are like suggestions. So it's very similar to, in case you know about ecology, very similar to the Treaty of Paris and the Protocol of Kyoto. So the Outer Space Treaty would be like Kyoto, very authoritarian, while Paris is like very COSPAR, is like inviting people to comply, and is the issue of the bioburden. So to not burden outer space or Earth with, especially outer space, with material from Earth, which is our next idea, which is bioburden, is the amount of biological contamination that you can introduce in planetary missions. So you've got like a level. I mean, I think that it's I think that it was 10 kilograms of that you cannot take of biological material, taking into account in case of eukaryotes like strains and all of the laboratory equipment, to not burden space. And well, these are as I'm the new major of iGEM Space, of Space Village. There are like lots of things that have been going on since 2009. 64 directly iGEM projects that they work on all of this. And well, iGEM right now is that I'm going to do a lot of changes, and you're going to be able to see them very closely. So I invite you to see them very soon. We are going to focus on the biology of space, on the policy, and we're going to organize some kind of events. And this is one of the projects that I participated in. So it was creating plants that could biosense different compounds of interest. So in this case, it's a tobacco plant that at high concentrations of CO2, it would change colors to red, and with pest sequences, it would turn back. So, for example, in outer space missions, astronauts can know if there is a leak, in case the electrical biosensors are faulty, and it also can be turned in space. So we have done it with CO2, carbon monoxide, sulfuric acid, heavy metals, and in the future we'd like to do it with explosives. So this is like the sequence that we did. So we did with a promoter, after a high inducible promoter for the compound of interest that we were interested in. So when there is no CO2, the promoter does not inactivate and the plant does not turn red. And in case of Ruby, and afterwards the exact same thing with Ruby. And when there is high concentrations of CO2, the plant turns into red because the transcriptional unit is being activated. And when there is no longer high concentrations of CO2, the pest sequences of degradation of proteins turn back to green. And this is the exact same thing with EYGVP. And this is a thing that I said, everything that we do research on in Earth or for space can be turned back to Earth. Like, for example, the Velcro that was researched for the space shoots, or the GPS for satellites. Everything that we have done for space can be turned back to Earth, and biology is not an exception. And everything that we do is going to help us understand better our planet, give us a couple of years more to change all of the things that we are doing wrong, so people in low, especially my issues with low and middle income countries, which right now they are at the mercy of the elements with all of the issues that they have, and while northern countries are exploiting them, and the only issue, the main solution is for synthetic biology to thrive, and to get out of the lab so we can prosper as a whole, so they can overcome the issues of drafts, high concentrations of radiation with the issues of the ozone layer, the need of water, the need of the bad issues of the environment with high concentrations of metal in lixiviation and eutrophisation. So that's why we should do more research on space, and I really hope it's done. So that's why we, for example, we do research on radio-tolerant enzymes, closed-loop biomanufacturing, minimal agriculture, self-free systems, autonomous nodes, so anyone can do synthetic biology in the future when it's safely enough so anyone can do it, and much more. I mean, sky's the limit, and quite literally. So, I mean, I think that this is the end. And these are some acknowledgements of people that have been helping me in my journey in space. So we've got Sara Garcia Walde, Lucas Boldrin, that is going to be our speaker at the end of the month from IBIS, and Jaime Puig, which is a great friend of mine that is a PhD student at SNOLAB, and he's going to also give a talk about his work. And thank you all to my labs, to iGEM, and to the European Society of Synthetic Biology for everything that they are doing to foster a healthy environment for synthetic biology. So thank you so much, and this has been all. I hope that you have not been very bored with everything. And right now, if you want to give any, if you have got like any questions or anything, feel free to ask.
Just wanted to thank you for the presentation, Yassin, and also wanted to ask, probably as humans, we have, that's 50 plus years of exposure to space environments. And probably there are hundreds of astronauts, cosmonauts that were already spent, accumulated maybe a century of time in space. Do we have, I don't know, some data on the effects this exposure to the space environment produced over time to these guys, or it's too personal to, I don't know, to make it available to a wider public?
Right. Well, that is an amazing question, actually, because right now the NASA in their AWG groups are doing research on that, and I've been overseeing and helping in some. I mean, I don't work a lot with mammalian tissues, but the thing is that the data is there. They have seen that Alzheimer's can be boosted in case of space exposure. So they've seen that lots of astronauts start developing early onset Alzheimer's and without having any predisposition. So the disruption of the cell membranes is heavily enough in case of the brain that it can have everlasting effects after coming back to Earth and after a couple of years. So they also have seen that on female astronauts, lots of them have had their menstrual cycles cut down to the point of having early menopause at super early ages. I think that one of the I think it was a Russian cosmonaut that had, oh no, it was an American that started having irregular cycles at the age of 30, and by 35 she did not have any more menstrual cycles, which was one of the main issues. And they have seen that the main issue is not radiation. Well, of course, cancer is very the high rate of cancer is way more higher in astronauts rather than in humans, especially skin cancer, which is way more aggressive because they have been exposed to this kind of radiations. But the main issues is that the disruption of these membranes coming by this microgravity comes from, especially for neurodegenerative diseases and in menstrual cycles. So the data is there. The data is available to the public. If you want, I can send you, Radio, or anyone in the audience some papers about that, because it's something that is very widely produced. But of course, the importance of knowing the effects have been studied only, I think that it was 30 years ago, so we still need more data. So I don't know if if you have understood it properly. I mean, if not, I can send you a couple of papers because they are very interesting, especially divided into age groups because they've seen that also between races. For example, people coming from black ethnicities. I mean, astronauts are not very. I mean, black astronauts have not been a thing for the last until the last years, unfortunately. But they have been speculated that black astronauts should be more resistant because the DNA machine, the repair of their DNA and the membranes is supposed to be stronger, so they should be more resistant to space, especially since they are living in places near to the tropics, and those places are very close to space environments with the high concentration, with high levels of radiations, the drafts and such. So they should be at least be a little bit more resistant.
Thank you. If there's no other question.
I think that if there are not any more questions, I mean, if you want, I don't know if...
I have one more. Maybe you have some insights about the number of the iGEM projects that will explore space this year. Are there any already getting in contact with you?
I have done a little bit of research because the iGEM teams have started to, well, they have already chosen their tracks and their villages. So this year iGEM has got 14 teams for space, which is more than I anticipated because I thought that we were going to have like between 7 and 10. And I haven't been in contact with anyone. I'm going to launch the space village in I have still to prepare a little bit of infrastructure to launch it officially, and then we'll start making some mapping and seeing how it's going to happen. But I really hope that more people tend to do research on space, especially because the bad thing about space is that people think about space synthetic biology as a separate discipline, you know, as if you are doing oncology or as if you are doing agriculture or food and nutrition, when everything that you are doing can be applied to space. I mean, you can do agriculture in space, you can do health on space, you can do oncology in space. So I don't think that it should be counted as its own village, rather, you know, because I mean everything, because all of the villages are applied to Earth, so all of the villages could be applied to space. But yeah, I mean, I haven't been in contact with anyone yet. I really hope that that I have some connections with them in the future because I have read a little bit of their abstracts and such, and they're looking quite promising. But yes, I haven't been in contact with anyone yet. But they're looking quite promising.
Okay. So there will be work to do in this year. Okay, great. Thank you.
Of course, yes. And if anyone wants to collaborate in the future, wants to learn more about space, I mean, I'm here 24/7, mostly with my network. So if for anything, I'm here to help. So I think that if there are not any more questions, I don't know if Davide wants to step in.
Yeah. Yes. Okay, so if there are no more questions, I would just thank you, Yassin. It was really an interesting talk, and I invite the audience to check our EUSynBioS page on Luma for the next events of the series focused on space that Yassin has been organizing. And yeah, we are very excited for the speakers coming up. I hope to see you in the next seminars.